All right, so today we're going to be talking a little bit about the patterns of inheritance. So we just went over a little bit of a review about variation, genetic variation. And so now we're going to try to think about, well, how does this variation really show up? In past classes, we've looked at it in a very simple way. We've considered, oh, there's some recessive traits, there are some dominant traits, and that's really how everything works. But that's not really true, like everyone is sort of a mix, a gradation between their two parents. And so how do these traits show up? Are there different patterns of inheritance? That's the thing that we're going to look at today. So, people used to think of genetics like paint, which is why I have this picture here. Two parents mix their genes, and the offspring falls somewhere in the middle. So you have blue paint and yellow paint, and then you get green. But this fails to explain a few things, like sometimes there are traits that skip a generation, for example. You might be short and your parents might be tall or something like that. Or there might be covering up. And so we're going to look at, there's this monk named Gregor Mendel that did an experiment on pea plants. And he was growing peas in Austria, and he sort of discovered this idea about how these organisms could pass on traits in some ways. So here is the example that we're going to take a look at with Gregor Mendel. So in his experiment, he had two plants. He had purple flowering pea plants, and then he had white flowering pea plants. He bred them together. Here, when we identify the pea generation, we're looking at P here. P is the parent. And so he had these purple flowers and these white flowers, and then when they combined, he bred them together. And then all of the plants had purple flowers. Like, that's pretty interesting, right? So the purple flowers and the white flowers together, and they all had purple flower babies. That's weird, because he expected to see something in between. He thought that there would be a mix of purple and white, some sort of a rosy color. That's not exactly what happened. Then he cross-pollinated these purple flowers. So he allowed the purple flowers to mix with other purple flowers. And what he ended up getting was a ratio, a different set of plants. He got 705 purple flowered plants out of 1,000. And he got 224 white flowered plants. And so this is weird to him that it skipped a generation. These white flowers didn't appear in this first generation of plants, but it did appear in the second one. And so Mendel had a theory for how this occurred. So his theory was that these traits were sorted according to an independent assortment. And so they were sorted independently from each other. So you can sort of tell this by, he eventually theorized that these purple flowers had something called dominant traits, which we've seen before. These are the capital P, capital P. These white flowers have recessive traits, lowercase p, lowercase p. And then when they're combined, you end up getting all flowers that are one capital and one lowercase. And that would happen if you did a Punnett square here. We could do one, but I think you all can kind of figure this out with two capitals and then two lowercase. And then every single child is going to be a capital P and a lowercase p. And so that's what actually ended up happening. All of the flowers here were one capital and one lowercase. But what happened was that when these one capital, one lowercase, or heterozygous flowers were mixed together, you ended up getting a larger variation of what those plants would turn into. And so you have this different variation that Mendel saw. And so the big takeaways from Mendel's pea plant experiments are as follows. One, some alleles are dominant and some are recessive, which means some of them are masked. That's what it means to be a recessive trait. A key misconception that people often think is the recessive appears less often. That is not the case. Recessive just means it gets masked. You might have all recessive traits in your population. That's totally fine. However, there are some traits that are dominant, some that are recessive. The two alleles separate from each other when the gametes are formed during meiosis. And so when we do meiosis, we'll talk about that a little bit later, but the traits independently assort. So you get one capital P, one lowercase p. These are independently segregated from each other from maybe a parent that looks like that. The third is that alleles for different genes move freely and separately from others during gamete formation, which means the same thing that we talked about. These alleles become separated from their parents. And so that's an important thing to remember as we talk about Mendel's and his laws. But this is not true of all human traits. We don't always follow the dominant and recessive rule. For example, hair. Our hair does not just get masked by one of our parents. It might not look exactly like either of our parents. Eye color, for example, is not always the same. height is certainly not one of those things that we get exactly from one parent or another. And so these things cannot be explained by simple recessive dominant rules. And so we're going to try to think about how these would occur. So there's tons of exceptions to this. There are things with 3 phenotypes, like hair. Hair has more than one phenotype. There are things with more than 3 phenotypes, like height. There's tons of different heights that you can get in the human population. There are things with more than 2 alleles, like blood types. And then there are things that are sex affected, as in males, the male sex is more likely to get one than the other, like hemophilia. And so we're going to look at a few different examples here. We're going to look at examples of incomplete dominance, things called multi-allelic traits, sex-linked traits, and then we're going to think about pedigrees, and then we're going to go through each one of these things and try to answer some questions related to them. So the first idea is of incomplete dominance. Incomplete dominance. So this is what Mendel thought. Mendel, in his experiment, thought purple and white together. He saw that this only led to purple plants because this was the dominant one. You got capital P, capital P. Here you got lowercase p, lowercase p. And so you ended up, when you combined them, you only got purple. However, we did it with a different plant. We did it with snapdragons, a different type of flower. And here, when you mixed the dominant individuals, the red flowers, with recessive individuals, what you ended up getting was one that showed both traits combined. You got pink flowers when they were heterozygous. So the heterozygous one showed up totally and completely different. So this is incomplete dominance. In this section, we call it incomplete dominance, where the neither of them are going to show up totally completely, but we're going to see a blend of them. Complete dominance, incomplete dominance. And so this is the example of what the Punnett square might look like. In the example of complete dominance, if you have a heterozygous individual, they're going to show up as the dominant trait. They're always going to be the dominant one. In incomplete dominance, if you have a heterozygous individual, they'll show up as a mix between the two. The next thing we have is an example of codominance. This is similar, however, it's different in that you can see both alleles. So in this case, when you have the incomplete dominance example, is that you show both the white trait and the roan trait here, the brown trait, and you end up getting brown dots on a white organism. And so this is codominance, where instead of some mix of colors between the two, so maybe in our last example of incomplete dominance, when these two combined, you would see like this type of color that's a little bit different. Instead, what you see is both of them appearing at the same time. So codominance here is when both alleles are visible. An example of this is something we may have talked about before, something called sickle cell anemia. Sickle cell anemia. And so this is where you have the normal individual, or the dominant one. The carrier shows some cells that are these sickle cell shaped, but not all of them. So some of them are like that. And then you have the diseased one, which is totally recessive, and then you see all sickle cell shapes. So this is an example of codominance, where you can see that if you have the trait for sickle cell, then some of your red blood cells in this instance will be shaped like a sickle. Okay, so we're going to practice. There's 2 questions here, and I'm going to pause right here to see how you guys do on those. The next thing we have is an example of multi-allelic traits. This gets a little bit complicated, but essentially multi-allelic traits are when you have more than 2 possible alleles for an example. And this can include, the biggest example is often blood type. I'm going to move myself over to this corner. I think that's probably best. So blood types is the sort of biggest and easiest example that we go to when talking about this. And so you may know that you can either have A blood type, B blood type, combination of the two, or O blood type. So there's lots of different blood types that you can possibly have that are related to different alleles that you get coded for. This is a word that you may have heard of in relation to COVID, but they're called antigens. So antigens are really the things that related to antibodies. They are the things on the surface of your blood cells that give you essentially the blood type that you're looking for. So you have 3 different types of alleles that code for blood type. One is that you have IA. This codes for the A antigen. So if you have IA, you're likely to have the A blood type. You have at least one of the alleles for this blood type. If you have IB, you can have the B blood type. You have one of the alleles for the B blood type. The lowercase I is what we will code for no antigen. In this case, usually that means you get some part of the O blood type. And so we're going to look at exactly what this means in a second here. So these are the different blood types that you can have. You can either have group A, you can either have B, you can either be AB, or you can be O. So these are all the different types of blood types that you can have as a human being. I, for example, am a B positive. And they're related allele combinations that would lead to them. So in this case, if you have two A's, you end up with an A blood type. That seems pretty obvious. In addition, if you have an A and the lowercase I, which gives you no antigens, this also will mean that you have an A blood type because these things code for the antigens. You can see here that as long as you only have these A antigens represented by this circle over here, then you'll end up with an A blood type. And we can see something similar would happen with B. AB is different, however. In this case, you have the alleles that code for IA and IB, and those will give you both antigens. So you can see that you have both the B antigen and the A antigen going on over here. And then finally, the only way to get O is if you have lowercase I, lowercase I. This is the only way to get an O blood type. So the rarest blood type of all is the O blood type. O negative is the rarest blood type. And so this is how you get this O blood type, is that you can only have this set of alleles. If you have any other combination, you're going to end up with a different blood type. And so this is why when you talk about the universal donor, which is the O negative blood type, that's a rare one. So this is a good chart to have under your knowledge, understanding these relationships. We're going to try a few multiple choice questions that are related to this. I'm going to put them up on the screen right now. Okay, so we got that done. These are those multiple choice questions, but I wanted you to have the chart. The chart really helps with figuring this out. Okay. The next thing we're going to look at is sex-linked genes. Sex-linked genes. So this is the gene. Well, this is an example of colorblindness. So if you're colorblind, you will not be able to see the figure 8 that's going on in here. The figure 8 is going on in here. But if you are not colorblind, then you will see a figure 8. This is an interesting test. So if you are colorblind, you'll see some information there. So this is kind of what it looks like when people see red in regular vision versus colorblind vision. And what's interesting about colorblindness is that men are 13 times more likely to be colorblind than women. If you have the Y chromosome, at least, I'll show you that you're 13 times more likely to be colorblind. And so what does this mean? How do we, why is that happening? And so what we've essentially discovered is that because of a chromosomal difference, and you may know that if you're born with the male sex, then you have an XY set of chromosomes. That's your 23rd chromosome, your sex chromosome. And females have XX. And this is a very binary understanding. There's more than these amount of chromosomes. Some people are born with XYY, some people are born with 3 of each chromosome. But in this very binary understanding of male and female, you are either born with the XX or the XY. And the issue is that, well, some of these traits, like colorblindness, is linked to these chromosomes, which means that you're more likely to have them if you are of a particular sex or born with a particular set of chromosomes. And so this is essentially how sex is determined. All egg cells have X cells. So you can see that from the female, you get 2 X chromosomes. And from the male, you get an X or Y. And if you do a Punnett square, it's fairly simple to see that 50% of the time you end up with 2 X's, 50% of the time you end up with XY. And so this is how generally sex is determined initially. Obviously that can change throughout your lifetime and different things have different effects on them. But traditionally this is what people thought of as the assignment of sex. And so the sex-linked chromosome is essentially that the Y chromosome can carry, the Y or the X chromosome can carry some certain traits. In this example here, there are genes found only on the X chromosome, which are called X-linked traits. And so those genes and those traits end up being more common in a certain gender. So here's an example here where you can see what's going on. Here's the example of the fruit fly color. So in fruit flies, the gene for eye color is found on the X chromosome. So red eyes are dominant to white eyes, and the Y chromosome has no gene for eye color, which means what ends up happening is that you see a difference in relationships between fruit flies based on their gender. So we're going to try to do a quick Punnett square here, where you have the red eye female fruit fly. So we can do X capital R and X lowercase r with a red-eyed male fruit fly with X capital R and Y. So we're going to Y. We're going to do a Punnett square there. See how that works for you. Pause it right here. And in the end, you end up getting a Punnett square that looks very similar to this, or it should look like this. And so they're related phenotypes, the possible phenotypes that you can get. You can see this one, you get red. This one, you get red. This one, get red. And this one, you get white. And so that's the females with red eyes are 100%. You also have to consider sex here. So if you have X capital R, you're going to be females with red eyes. And then you get males with red eyes. And you get males with white eyes as this last one. So in this result, because of its X-linked nature, every female that is born will have red eyes. 100% of the time, all females have red eyes. And 50% of the males have white eyes because, there's two options here. And so that's how you get this particular probability as a result. So hopefully that made sense because we're going to try a few examples of this. All right. So this is going to be the end of this part of the lesson. Hopefully that made sense, and hopefully some of those questions made sense too. And then after this, we'll get back into the rest of our stuff. Thanks, everyone.